control transgenic plants, figures 1c and 1d. We specifically compared PHB expression levels in vector-controlled transgenic plants, MIM165, PHB1D, and STTM165-166-48, and found that PHB expression in STTM165-166-48 was a little lower than that in PHB1D but much higher than that in MIM165, figure 1c. In summary, the above data strongly support the conclusion that STTM165-166-48 inhibited the activities of MIR165-166 and caused an increase in the levels of 5-HT-ZIP3 transcription factor genes, leading to a series of developmental alterations. The effect of STTM165-166-48 is comparable to the gain-of-function PHB1D mutation, but much stronger than the IPS1-based MIM165 in both phenotypic severity and the upregulation of the mRNA target genes. The optimal length and the secondary structure of the RNA spacer between the two mRNA binding sites. In animals, mRNAs targeted by mRNAs usually contain, within their 3' untranslated regions, multiple mRNA binding sites that are bound by either one mRNA or different mRNA species. Bartle, 2009. The distance between the mRNA binding sites is important in LET7-mediated translational repression of Lin41 mRNA in Caenorhabditis elegans. Vela et al., 2004. To explore how the length of the spacer could affect the functional blockage of MIR165-166 by STTM165-166, we designed spacers with different lengths, 8, 31, to 48 nucleotides, between the two MIR165-166 binding sites and introduced them into plants, figure 2a. The severity of phenotypic alterations was used to score the transgenic plants for the effectiveness of the STTM. The length of the RNA spacer between the MIR165 and MIR166 complementary regions is crucial for STTM165-166's function. A. Diagrams of the STTM structures with varying lengths of the spacer region. Orange indicates the spacer region and the spacer sequence. Blue indicates the bulge sequences in the mRNA binding sites. Red indicates the nucleotides that are different between MIR165 and MIR166. B. The phenotypes of representative STTM165-166 transgenic plants with different lengths of the RNA spacers at different developmental stages. Rows 1 to 4 represent plants at days 4, 8, 15, and 30, respectively. NT. Nucleotides. We mainly examined the morphology of cotyledons, true leaves, and overall plant architecture. After germination, the STTM165-166 transgenic plants with a 48 nucleotide spacer, STTM165-166-48, exhibited altered cotyledon morphology such that the cotyledons were spoon-shaped, figure 2b. During later stages, the STTM165-166-48 transgenic plants always showed the strongest phenotypic alterations in the shape and polarity of the true leaves as well as overall plant architecture, figure 2b. STTM165-166 plants with 31 nucleotide spacers, STTM165-166-31, exhibited less severe phenotypes than those of the STTM165-166-48 transgenic plants, while the transgenic plants with an 8 nucleotide spacer resembled plants transformed with the empty vector. Given that STTM with a longer, 48 nucleotide, spacer had better efficacy than that with a shorter, 31 nucleotide, spacer, we further extended the 48 nucleotide spacer of STTM165-166-48 to 88 and 96 nucleotides. See supplemental figure 1a online. While retaining the stem structures, see supplemental figures 2d and 2e online, formed by the spacers between the two mRNA binding sites. Compared with STTM165-166-48, STTM165-166-88 had increased efficacy. See supplemental figure 1b online. But a further increase in the spacer length to 96 nucleotides did not improve the efficiency significantly. See supplemental figure 1b online. One potential function for the spacers used for STTM might be in the stability of the expressed STTM transcripts. We thus performed a thermodynamic stability analysis of the potential RNA secondary structures for STTM-165-166 RNAs with different lengths of spacers. See supplemental figure 2 online. As suggested by RNA folding analyses, the structure of STTM-165-166-8 RNA was predicted to be rather unstable with a DG of -11.7 kilocalories per mole. STTM-165-166 structures with longer spacers were predicted to be more stable with a DG lower than -20 kilocalories per mole. The lack of efficacy of STTM-165-166-8 might be attributed to its low stability. However, the difference in effectiveness between STTM-165-166-31 and STTM-165-166-48 could not be simply explained by their thermostabilities because the DG values of STTM-165-166-31 and -48 were even slightly opposite. See supplemental figure 2 online. Further analysis of the folded RNA structures showed that STTM-165-166-48 formed a stable stem region that did not exist in STTM-165-166-31, leaving the two STTM modules as dangling ends that perhaps would make it possible for Mir 165-166 to dock onto the STTM. See supplemental figure 2 online. The stem region of STTM-165-166-48 may play a role in stabilizing the STTM transcripts and explain the difference in effectiveness between STTM-165-166-31 and STTM-165-166-48. Extending the spacer length from 48 to 88 or 96 nucleotides while retaining the stem structure increased the STTM thermostability and consequently the efficiency in the reduction of the target mRNAs. See supplemental figures 1 and 2 online. Additionally, we randomly mutated the STTM-165-166-48 in the spacer region without changing the spacer length to generate STTM-165-166-48 MUT, in which the stem structure in the spacer was altered. See supplemental figure 3 online. Compared with STTM-165-166-48, the proportion of STTM-165-166-48 MUT transgenic plants with strong abnormal phenotypes was reduced approximately from 30 to 20%. Taken together, these analyses suggest that the thermostabilities and the secondary structures of STTM transcripts, especially the stem region formed by the RNA spacer between the two STTM modules, might contribute to STTM efficacy. STTM-directed functional blockage of Mir 165-166 occurs through the reduction of Mir 165-166 levels. Recent studies indicated that TM also triggered reduction of the targeted mRNAs. GU et al., 2010. Tedesco et al., 2010. To investigate the specific mechanism and the effectiveness by which the STTM inhibited Mir 165-166 functions, we conducted mRNA gel blotting analysis to determine the levels of Mir 165-166. MIM-165 plants, which were included as a control, showed a modest reduction in Mir 165-166 levels as observed before. See supplemental figure 4 online. Tedesco et al., 2010. Consistent with the stronger developmental defects caused by STTM 165-166-31 or -48, Mir 165-166 was almost undetectable from STTM 165-166-31 and STTM 165-166-48 transgenic plants. See supplemental figure 4 online. Plants transformed with vector alone or STTM 165-166-8 had much higher levels of Mir 165-166 than STTM 165-166-31 and STTM 165-166-48 plants. Figure 3a. The reduction in Mir 165-166 levels by STTM 165-166 was very specific. Mir 168 was not affected by STTM 165-166, figure 3a. Further examination of miR-165-166 target genes in the corresponding transgenic plants revealed that all 5 targets were upregulated significantly in STTM-165-166-31 and STTM-165-166-48 transgenic plants, but not in the empty vector control or the STTM-165-166-8 transgenic plants. See supplemental Figure 5 online. The above data strongly support the conclusion that STTM-165-166 triggered a reduction in miR-165-166 levels and activated 5-HT-ZIP3 transcription factors in the transgenic plants, leading to a series of developmental alterations. STTM-165-166 triggered drastic reduction of miR-165-166 levels. A. Representative RNA gel blotting to detect STTM-165-166, miR-165-166, and miR-168. U6 served as an internal control. NT. Nucleotides. B. STTM-165-166-48 was unable to affect the migration of miR-165-166 on a denaturing polyacrylamide gel. C. Copy numbers of small RNA reads in vector control in STTM-165-166-48 by deep sequencing. To determine whether the failure to detect miR-165-166 by RNA gel blotting was due to the artifact of in vitro sequestration of miR-165-166 by the 108 nucleotide STTM structure during electrophoresis, we generated the 108 nucleotide STTM-165-166-48 RNA transcripts by in vitro RNA transcription and subjected them to in vitro miR-165-166 binding. After ensuring full annealing between radiolabeled synthetic miR-165-166 and non-radiolabeled STTM-165-166-48 transcripts by examining the altered migration on a native gel, we resolved the annealing products on a denaturing polyacrylamide gel without a heat denaturing step prior to sample loading. It was clear that the STTM-165-166-48 RNA transcripts were unable to sequester the radiolabeled miR-165-166 to the expected position, approximately 108 nucleotides, or significantly alter the migration of miR-165-166 in the denaturing gel, Figure 3b. Therefore, our results suggest that the observed reduction in miR-165-166 levels by STTM-165-166-48 was not likely due to the sequestration of miR-165-166 during gel electrophoresis. To further confirm the reduction of miR-165-166 levels in vivo in STTM-165-166-48 plants, we constructed small RNA libraries using the small RNA fraction isolated from the aerial parts of 3-week-old seedlings of vector control in STTM-165-166-48. The abundance of small RNA reads of each sample was normalized to 9 million, and the read numbers of miR-165, miR-166, miR-168,